develop a system of classification. Background. With so many different plants, animals, and microbes, how can they be organized and named? In order for scientists to study and communicate about organisms in a systematic way, they must classify them according to characteristics they have in common. This is quite a task considering there are over 1.5 million species. Taxonomy is the name given to this science of classifying organisms. Aristotle was one of the first to attempt some type of organization system, dividing organisms into plants and animals. This may not seem to be a very complex job, but even dividing organisms into plants and animals can be challenging since there are some one-celled organisms that share characteristics of both. Since then, scientists have broadened these two divisions into 5 large categories or kingdoms, further dividing and subdividing them based on an organism's shared and unique characteristics. Scientists then named these divisions as follows: kingdom, phylum, class, order, family, genus, and species. The scientific name for each organism includes its genus, which is capitalized, and species name. For example, humans are Homo sapiens. But who chooses what particular characteristics will be the ones that define a category? How different would things be classified if some other characteristics were defining one? In this lab, you will have a chance to experience the difficulty of the science of taxonomy. Problem: how could differing characteristics of seeds be used to form a system of classification?
Awesome. Thank you so much, Alicia. Sarah, what I'm going to do is at the end of class, I will scan these and upload them for you so that you will have a copy of them. So what we are going to do is, if you turn to page 64, we are going to go ahead and complete the lab. I am going to change the view of my camera so that Sarah and everyone else can go ahead and see my desktop. Just give me 1 second here. Okay, and you should see a white sheet of paper underneath on my desk. I will caution you, I cannot read what is written in the text chat right now, so if you are trying to get my attention, please raise your hand. I can hear that, but I can't see my screen right now. So what we are going to do is we are going to start with today's lab. Alicia, you have a question?
Sorry, I cannot see your white paper or I can't see it. You know how you said you changed your perspective of the camera? I can't. Okay, now I can. I'm sorry. It must have frozen.
Okay. Hold on 1 second again. Okay, perfect. There we go. Now you should be able to see it, hopefully. Okay, so part 1. Find the seeds in your lab kit and empty them onto a clean, flat surface, preferably white. The reason they say white is it's easier to see. I do not care what color you are looking at here. Okay, so I'm going to open up my baggie of seeds. I'm going to pour them onto my white paper, on which you would be able to see all of the seeds clearly. Make the taxonomy chart similar to Figure 2 and title it Part 1. So I went ahead and I have already completed that, and I'm going to go ahead and switch my papers, and you should be able to see my taxonomy chart. Now, I do know that you will not be able to see it perfectly. However, just to give you an idea, here is my top one that says seeds. Place the word seeds in the topmost circle count out the number of seeds and put that number under the word seeds. So you are going to take the seeds that we just dumped out, you are going to count them and put them into the seed pile. So if you have part of a seed, that does not count for anything. Only use whole seeds. So go ahead and everyone take a minute to count your seeds. Once you have your total number of seeds, click yes on your screen. Now, Sarah, since you are not able to do the lab with us, just go ahead and click yes for these. This just lets me know when everyone is ready to move to the next step. Okay, fantastic. Make sure you write that number down somewhere. The next step is you will be dividing and separating the seeds using varying characteristics. The first characteristic is size. Divide the seeds into two piles, large and small, and if you would like a third pile for medium, go ahead and do so. So what I'm doing, and I'm actually going to hold my paper up for a second, is I now have on my paper seeds, and I had 70 total seeds, so I'm going to write 70. And then I have a large circle and a small circle. I now am going to take my seeds and I'm going to divide it between those two circles. So all of my large seeds will go over here. My small seeds will go on the other side. Now just by looking at mine quickly, I know I have more large seeds than small seeds. So I only need to count my large seeds and then subtract the number from 70 to get my small seeds. So separate your seeds, large and small. Actually, I was wrong. I had more large seeds than small seeds. Same thing, when you are done with that, click yes. On the poll. We're going to use these polls often in class. It just keeps us together. Awesome. So now on my paper, I have 70 total, 42 large, 38 small. Okay, the next step is place the words large and small. Oh, we just did that. Next is to divide into shape. For the pile of small seeds, divide them into their different shapes. This is where you can divide them how you want. Round, kidney-shaped, oval, etc. And you're going to split just your small seeds into those categories. Then you are going to split your large seeds. So if we look at my paper, I'm going to split my small seeds, and I am going to do one circular and one oval. So I'm going to take my oval seeds and put them there, and I'm going to take my little circular ones and put them here. Go ahead and do this for both your large and your small seeds. And then count them. Okay, and same thing. Once you are done with that step, go ahead and click yes. Awesome. Okay, so now, oopsies. Now my taxonomy chart looks like this. Let me back it up. And lastly, we are going to use color differences to divide each of the piles of seeds into their various colors. So you are going to list the colors under the shapes. So for example, my large circle ones, I only had brown. So all of them are going to go there. My large oval ones, I had white. I had yellow. And I have brown. So I am going to add this to the bottom of my taxonomy chart, and I am going to count them out. So go ahead and do that. While you guys are finishing that step up, I'm going to go ahead and put my data up on the whiteboard. Okay, when you are done, go ahead and take a look over at the whiteboard. And this is my data that I got from the lab. Your data may look the same. Your data may look completely different. But either way, this was my data. Now what I'm going to do, Sarah, is I am going to download this sheet as a PDF so that you can use it when you are writing your lab report today or before next class. And what I would like you guys to do is just the same thing. Go ahead and click yes when you are ready to move on. Okay, fantastic. So let me download this for Sarah. And I will go ahead and I will put this data in Moodle so that she can refer to it and any of you can refer to it if you need to. So that is the end of part 1 of the lab. Now for homework this week, you guys are going to finish this lab doing part 2 and writing a lab report. Okay, Sarah just asked a good question. How will I be able to find this later in Moodle? That's a great question. I believe all of you finished the orientation assignment in Moodle, so you've all been into Moodle. If you scroll down our class page, so you're going to click Biology Lab from your courses, and then you will see where you had the orientation. If you scroll down, it will say Week 1. And it will have, I believe it says classification. It will give you a little bit of information about the class. It will have all of the PowerPoints for today's class in it. And it will also have a recording of today's class, as well as your homework assignment. I'm going to add this up there as well, Sarah. So I will label it Classification Lab Data so that you can go ahead and gain access to it, or anyone who needs to review the data can see it. If you were able to complete this activity, I would love for you to use your data and turn it into me. If you were not, for whatever reason, you certainly can go ahead and use my data. What I'm going to do now is we are going to actually go into Moodle, and I wanted to show you a couple things, so I can show you that as well. So I'm going to turn my camera off, and we are going to go into Moodle. Okay, right now you should see the Moodle webpage. This is our class. If I scroll down, it says Animal Classification. It has Spot the Difference, which is the giraffe activity we did. It has Biology Lab Safety, which was that PowerPoint. Taxonomy PowerPoint, which we looked at. Kingdoms and Domains, which we did not look at yet, and I'm not sure we will have time to. It says Homework Week 1. Now, in order to do your homework this week, you are going to click on Homework Week 1. Your homework for week 1 says upload your lab report here. You are going to, upload your lab report into Moodle. But you might be saying, well, that's great, but I don't know what you want for a lab report. In that case, let's go back real quick. Right underneath your homework week 1, I wrote lab report. In science, there are some normal things covered in lab reports, but everyone has their own way of doing lab reports in their own format. What I would like you to do is click on where it says lab report and a PDF will come up. This is your lab report guideline. Now, I have loaded this lab report guideline into Moodle so that we can look at it easier. So take a look at your PowerPoint screen. And this is what I would like for a lab report. Your lab report should be typed. You should have a title. In most cases, your title will be the title of the lab. So in this case, your title is going to be Lab 13, Classification. You are going to make a hypothesis if needed. Now, today's lab, there is no hypothesis to be made. So you are going to skip this. Materials, you are going to list all materials we used. All meaning like everything. So for this lab, we would have our bag of beans. We would have a pen or a pencil. We would have a piece of paper. Okay, you need to make sure you have everything listed there. If you use water in the lab, that goes in the materials. You are going to list your procedure. You can do your procedure either in a paragraph form, or you can write your procedure step by step. I do not have a preference whatsoever, but the way a procedure works is I should be able to print off just your procedure and your materials, hand it to my son, and say, do this lab. And he should be able to accurately complete the entire lab. So that is how specific your procedure should be. Think of it as you are going to hand it to your brother, your sister, and say, please finish this lab for me. Then you are going to write your results. Well, your results are your data, either your numbers or observations. For today's class, your results are going to be this picture. Now, you can scan this picture into your lab report. You can take a picture with your camera and insert a JPEG. You can draw it on the computer if you want. But this is your data. These are your results. For part 2, you are going to do something similar. Those are going to be your results or your data for part 2. Then you are going to include any questions in the lab report in your results section. So, for example, on page 66 of this lab, there are 6 questions. You are going to answer those 6 questions right underneath your data. The easiest way to do this is to write 1, period, and write your answer. If you want to make it into a paragraph to sound nice and neat, go for it. But you do not need to. And then you are going to write a conclusion section. Conclusion is, why do you think something happened? Well, this lab is pretty hard to write a conclusion about, so there really is no conclusion for this lab. Conclusions, when we are actually doing an experiment, are going to be easier to write. So in this lab, you are not writing a hypothesis, and you are not writing a conclusion. But you are going to complete part 2 of the lab and write a lab report for your homework this week. You then are going to upload it into Moodle by Wednesday at 9 a.m. I will grade it. I will send you a note back saying, good job, next week make sure you list all of your materials, you forgot a piece of paper. Or maybe my comment will be, your procedure is not clear enough. Please go back and clarify step 15, or whatever step it is. Okay, anyone have any questions about how to write your lab report? You also need to make sure that your name is somewhere on your lab report. Any questions? No? Yes? No? Perfect. Okay. Excuse me. So what we are going to do is we are going to look at a different PowerPoint now about kingdoms and domains. We're not going to get through all of the kingdoms right now, but we are going to take a look and start going through them. So take a look back at your PowerPoint screen. We are going to look at kingdoms and domains, and I called this PowerPoint Parade Through the Kingdoms. Okay, there are many kingdoms of life. Biologists have organized living things into large groups called kingdoms. Biologists grouped the organisms into 6 kingdoms based on RNA and DNA sequencing and similarities, as well as cell type, meaning is it a prokaryote or a eukaryote, cell walls, body type, are they unicellular or multicellular, and then nutrition, are they autotrophs or heterotrophs? Here are the 6 kingdoms of life. Take a second and look at these. These all probably familiar to you. I'm guessing you have all heard of the 6 kingdoms of life. There are 3 domains in life. The domains in life are based on a phylogenetic tree based on rRNA sequencing that demonstrates the division of all living things into 3 broad categories. Those 3 broad categories are bacteria, eukarya, and archaea. The domain that's thought to be the oldest is bacteria, which is composed of the organisms in the kingdom Eubacteria. Archaea is the second prokaryotic domain and is also composed of a single kingdom, Archaebacteria. The third domain is eukarya. Eukarya contains all 4 of the eukaryotic kingdoms: Animalia, fungi, plantae, and protista. So the domains in life are broader than the kingdoms. First, we are going to look at the domain bacteria. The domain bacteria contains a single kingdom, Eubacteria. Some scientists call this kingdom bacteria. Bacteria are prokaryotes. They have no internal compartments, and they are found in practically every environment on Earth. Bacteria have strong cell walls made of peptidoglycan, which is a web-like molecule complex made of carbohydrate strands cross-linked by short peptide bridges. And here is a diagram of characteristics of a bacteria. You can see the cell wall, the cytoplasm, the nucleoid, and the ribosomes. There are lots of kinds of bacteria. Some bacteria can cause disease, while others are used by humans to process foods. Bacteria are used to control agricultural pests, to produce chemicals, and perform genetic engineering. Some bacteria obtain energy from inorganic compounds, such as hydrogen sulfide, ammonia, and methane. Some bacteria are photosynthetic and are found in ocean and freshwater ecosystems. Some are heterotrophic. These bacteria are able to live without oxygen. Heterotrophic bacteria are also very important decomposers. In the domain Archaea, there is a single kingdom, which is Archaebacteria. Archaebacteria are prokaryotes that diverged very early from bacteria. They are more closely related to eukaryotes than to bacteria. Some characteristics of Archaebacteria are a cell wall and a membrane. The cell walls of Archaebacteria do not contain peptidoglycan, as the cell walls of bacteria do. Archaebacteria contain lipids very different from those of bacteria or eukaryotes. The gene structure and translation. The ribosomal proteins of Archaebacteria are very similar to those of eukaryotes and are different from those of bacteria. Some kinds of Archaebacteria are methanogens, the Archaeobacteria obtain energy by combining hydrogen gas and carbon dioxide to form methane gas. Methanogens live deep in the mud of swamps and are poisoned by even traces of oxygen. Extremophiles are a group of extremophiles called thermophiles, live in very hot places. Haliophiles live in very salty lakes that can be 3 times as salty as seawater. Other extremophiles live in very acidic places or under enormous pressure. And another kind of Archaebacteria is non-extreme Archaebacteria, and these grow in the same environments as normal bacteria. So basically out in the Earth's surface, in places like your yard, just like regular bacteria thrive. And that is where we are going to stop for today. I appreciate you guys participating in class. If you start on your lab report and you have any questions at all, please give me a call and let me know. There is absolutely no reason for you to feel like you can't contact me. If you would rather email me, email is fine as well. Sarah, I will work on that lab kit for you. And Gabrielle, I will work on a label for you. Look for something from me this afternoon, both of you. And Sarah, I will upload this diagram as well as the video to Moodle so anyone can access that. If you guys have any questions, I will stay on for a minute or two. Otherwise, it's 1:30. That is the end of our class for today. Reminder to make sure your homework is in by 9 a.m. next Wednesday. And I will see you all at noon Eastern Time on Wednesday. I hope you have a fantastic day and enjoy today's class.